Hi everyone, and thank you for taking the time to listen to my presentation today. My name is Kelly Balfour, and I am a graduate student at Queen's University. In this project, we investigated trait overlap within herbaceous plant communities to determine whether limiting similarity was more evident amongst positively co-occurring plant species. Niche theory dictates that species fill specific functional roles or niches as members of an ecosystem, and that they require a certain set of environmental conditions in order to survive. But because the natural environment is subject to resource fluctuations and limitations, an ecosystem cannot support an infinite number of species within a community. Plant communities are, therefore, competitive systems, within which individuals interact and compete with conspecifics to ensure their own survival. Measuring plant functional traits is a common practice to assess their resource requirements. These traits can impact plant growth, reproduction, and survival, and are thus good estimates of individual fitness. Functional traits refer to several whole plant, leaf, root, and seed characteristics, which are in turn associated with particular functions. In this diagram, I highlight some examples. Maximum height is related to lifespan, time to maturity, seed mass, and light capture. Seed dry mass strongly influences species dispersal and establishment ability. Specific leaf area is associated with light use efficiency, relative growth rate, leaf lifespan, and tissue nitrogen. And rooting depth allows for the interception of both water and nutrients. If we examine the full suite of functional traits possessed by this one individual, we can also identify potential competitors, as other individuals with similar trait combinations are likely to compete for common resources with this individual. Remember that common resources are also limited under natural settings, so we can expect the strongest competitor to persist in the environment while the weaker competitor is excluded. A community consists of numerous co-occurring species and fewer coexisting species. Co-occurrence refers to any species that are found together within the same area. However, they may only be found together for the time being and are not necessarily coexisting. Coexistence describes a long-term relationship between neighboring species. After competitive interactions for common limiting resources remove some species from the community, the remaining species can persist together for some time. Co-occurrence can also be positive or negative in nature. Positive co-occurrence describes species pairs that, when found together, rarely compete for resources, while negative co-occurrence describes species pairs that, when found together, compete intensely. Let's go over this with a diagram. Here we see three herbaceous or non-woody plant species: bromus, phleum, and taraxacum. Above each of these plants is a pie chart, which acts as a simplified representation of the resource requirements for each plant. In yellow, sunlight; in blue, water; and in green, nutrients. Of course, these requirements are more detailed and complex in real life, but let's pretend that those pie charts represent the conclusive needs and ecological niches filled by our three theoretical plants. We can see that bromus and phleum have very similar niches. Both require high light and nutrient conditions in order to persist. Their similar niches are also made evident by their traits, which aid in resource acquisition: large height, fibrous roots, and large leaf areas. When these two plants are found within the same area, they are likely to compete for common resources. Therefore, they form a negatively co-occurring species pair. Phleum and taraxacum, however, have different resource requirements, with taraxacum's pie chart indicating that it needs more water and less light and nutrients. Their different niches are once again made evident by their traits. Taraxacum is shorter, with a taproot system, and smaller leaves. When these two plants are found within the same area, they are unlikely to compete for common resources. Therefore, they form a positively co-occurring species pair. In a community containing just these three species, it's possible that they'd be able to survive using the resources present, and that they'd be able to persist together. However, species richness is much greater in natural herbaceous communities. The added species interactions within these communities highlight important environmental limitations. Limiting similarity describes a limit to the number of functionally similar species that can coexist in any ecosystem. This is due to competitive interactions for resources. It furthermore states that the coexistence of a species pair requires minimal niche overlap, so that the species will not compete intensely. Coming back to our phleum and bromus species on the left, their high niche overlap indicates that they are unlikely to appear together on account of limiting similarity. However, phleum may be found alongside taraxacum, as shown on the right, as the two have low niche overlap. As the environment can only support a finite number of species, limiting similarity is in accordance with classical niche theory. and yet evidence of limiting similarity can be difficult to obtain. Past studies of microorganism and animal assemblages have reported evidence of limiting similarity. And in 2019, my supervisor co-authored a study that identified temporal niche separation between coexisting bee-pollinated plant species in an old field meadow. This study adds to a mere 12% of studies that have found evidence consistent with limiting similarity in herbaceous vegetation, communities like old field meadows and grasslands. Although findings are rare in these communities, limiting similarity has been found in other assemblages and is expected under classical niche theory. So why isn't there more evidence? In this study, we wondered whether current analyses, which use a trait-based null model approach, were simply unable to detect these relationships. Typically, assessments of limiting similarity substitute coexistence data with co-occurrence data, which give you an idea of what species happen to occur together at the time of sampling, rather than what species exist together in the long term. These snapshots of community composition do not account for the temporary presence of negatively co-occurring species, whose niches tend to overlap greatly with their neighbors, as mentioned previously. Let's look at another diagram. If we look at the community on the left, two species have been identified as negatively co-occurring. Over time, and due to competitive exclusion, we can expect that community to look more like the one on the right, which consists of positively co-occurring species. Although negatively co-occurring species are not representative of long-term species coexistence, they are still included in traditional assessments of community composition. In the study, we assessed whether positively co-occurring species pairs, or positive pairs, had lower trait overlap than negatively co-occurring species pairs, or negative pairs. This result would suggest that positive pairs exhibit less niche overlap, as expected under limiting similarity, and this would also support the removal of negative pairs in modified assessments of community composition, which I'll talk about later. We extended invitations for collaboration to researchers in Canada, the United States, and Sweden, who added non-experimental study sites consisting of perennial herbaceous species. Our collaborators contributed plot-level presence-absence data, as well as any accompanying trait data for their species. We included 7 traits in our analyses: leaf area, leaf dry mass, leaf dry matter content, specific leaf area, leaf water content, maximum height, and seed dry mass. We performed pairwise co-occurrence tests to identify positive and negative species pairs, and measured the amount of trait overlap between each pair. We then compared trait overlap between positive and negative pairs, both within and across the study sites. We analyzed a total of 13 data sets. However, data for all 7 traits was not available for each site. Leaf dry matter content, leaf water content, and seed dry mass data, for example, were only available for 4 or 5 data sets, whereas specific leaf area and maximum height data were available for 12 data sets. Of these 7 traits, none were available for all 13 data sets. To assess trait overlap, we compared the mean trait distances of positive and negative pairs within each of the 13 sites, as well as across all 13 sites, using mostly non-parametric Mann-Whitney-U tests. Mean trait distances illustrate the difference between the least and greatest trait value between the pairs. Therefore, a large mean trait distance was indicative of dissimilar traits, while a small mean trait distance was indicative of similar traits. 3 results were possible for each comparison. Firstly, our null hypothesis was that positive and negative pairs may not have significantly different mean trait distances, indicating that trait overlap does not differ according to the type of co-occurrence. This result was not expected, as natural systems should limit the amount of similarity between positively co-occurring species. Secondly, positive pairs could have greater mean trait distances than negative pairs, indicating greater trait overlap by negative pairs. This result is expected under limiting similarity. Finally, negative pairs could have greater mean trait distances than positive pairs, indicating greater trait overlap by positive pairs. This would be possible if species required a common trait or adaptation for survival. For example, large maximum height may be necessary for light competition. These box plots illustrate the results of our tests when trait overlap was compared across all 13 sites. We found that mean trait distances between positive and negative pairs were only significant for specific leaf area, and that positive pairs had greater mean trait distances than negative pairs. This means that negative pairs have similar specific leaf areas, which is expected under limiting similarity. These box plots illustrate the results of our tests when trait overlap was compared within each site. 2 traits returned significant results. For specific leaf area in 2 sites, we found that mean trait distance was greater for positive pairs. For maximum height in 4 sites, however, we found the opposite result. Mean trait distance was greater for negative pairs, which isn't in accordance with limiting similarity. On this slide, I've included our findings for both specific leaf area and maximum height, providing an image to represent what those findings would look like for both positive and negative pairs. As expected under limiting similarity, positive pairs had different specific leaf areas, and negative pairs had similar specific leaf areas. Surprisingly though, positive pairs had similar maximum heights, while negative pairs had different maximum heights. Although this result is not in accordance with limiting similarity, it's still an interesting finding. Perhaps similar heights are reflective of a beneficial adaptation. For example, plants of different heights may compete more intensely for light, resulting in negative co-occurrence. There is some suggestion that positive pairs may differ from negative pairs, but a few considerations may strengthen these findings. Firstly, there are many different functional traits, but only 7 were considered here. So other traits like leaf nitrogen content, rooting depth, and biomass could be included in these analyses. Also, 5 of the 7 traits studied are leaf traits, so including more whole plant and root traits may be particularly insightful. For the trait data included in our analyses, we weren't able to transform them to meet assumptions of normality. And because our analyses use trait data from existing datasets, data for seed dry mass, leaf dry matter content, and leaf water content were only available in 4 or 5 of the 13 study sites. Collecting more trait data within each study site may reveal additional relationships that our analyses may have missed. Secondly, we acknowledge the potential role of environmental conditions on these data. Environmental data were not assessed as potential covariates in our analyses, and resource availability was not measured within each site. Trade-offs may impact competitive ability for some resources. For example, when nutrient availability is limited, light competition can be less intense. So should one resource be particularly limiting, competitive ability for other resources may decrease in effect. Regarding our analyses, if resource A was more limiting than resource B, for example, we could see more results consistent with the null hypothesis, that positive and negative pairs do not differ in their mean trait distances, particularly for traits related to the acquisition of resource B. Finally, traditional assessments of limiting similarity do utilize a trait-based null model approach. However, we first wanted to compare positive and negative pairs and quantify trait overlap between them. As traits were analyzed independently in our study, multivariate analyses may yield additional findings, and a null model would allow these data to be compared to expectations under chance. Regarding our initial question of whether limiting similarity was more evident amongst positively co-occurring plant species in herbaceous communities, we found that evidence consistent with limiting similarity is still rather limited. Specific leaf area was similar amongst negatively co-occurring species, suggesting that limiting similarity does exist within herbaceous vegetation, even if findings are scarce. In addition to limiting similarity, however, the similar maximum heights of positively co-occurring species suggest that additional factors, such as adaptations, may also influence community compositions. Ultimately, we will incorporate these findings into a larger publication, which will include a trait-based null model approach. The second question we will address in this publication asks whether the removal of negative pairs from assessments of coexistence will increase evidence of limiting similarity. Here we plan to compare the results obtained from a null model using a traditional approach, which includes both positive and negative pairs, as well as our proposed modified approach, which includes only positive pairs. We expect that this modified approach will reveal more evidence of limiting similarity from the same datasets, as negatively co-occurring species pairs are not representative of true coexistence. Our project addresses the lack of evidence of limiting similarity within herbaceous communities, and proposes a modification to the existing methodology. Over time, we may yet uncover and better understand the mechanisms contributing to species coexistence. Finally, I'd like to acknowledge the many people that made this project possible. My supervisor, Dr. Brandon Champ, Danielle Greco and Hannah Brazo, who worked on the R code for our analyses, our collaborators for contributing their data towards this project, and of course, I'd like to thank all of you for listening to my presentation today.